So welcome everybody to another StaPH-B call. I'm really excited about this particular StaPH-B call. Erin approached me, I guess it was a couple months ago, and wanted to present on some things because she'd been getting some, it sounds like, questions about some of the programs that we use, or that she was using, Trycycler and Unicycler, and kind of some of the, I think it excited me to bring back kind of the crux of StaPH-B and get back to kind of our meat and potatoes of, when we began this, we were all kind of learning about different software, different programs, things that we could use for analysis of data. And I think that with the past, year and a half to 2 years, we've had kind of a little bit of an interruption with the pandemic and with COVID just kind of taking over everything and all of our lives. So it's nice to get back to some different things sometimes, and to be able to kind of go back and look at some of the different software and programs that are out there for bioinformatic analysis. And so I hope that this call is going to be informative for you all, and hopefully maybe raise some questions and provide some answers as to what some of the differences are between some of these things. Before we get started, I did want to have one little kind of housekeeping announcement too. I know that Kelsey posted on the Slack channel yesterday about some updates that she's been making to the website. And so the StaPH-B website is going through some changes right now. We're trying to make sure that we have all of the things that we have created and that are available through kind of the StaPH-B loose organization that are available on the website and accessible to people. And so it's going to be going through some kind of updates and changes, and we're going to kind of continue to update it as we go forward. But I think that right now it's in its kind of like initial stages. And so we welcome any feedback. And I think that it should be exciting to check out. I've kind of browsed around it a little bit and taken a look, and it looks really beautiful. It's a very sleek layout. So look forward to any feedback that anybody has, and any sort of feedback that we can get on anything that you would want to see or anything that you would want added to the website. Please feel free to reach out either on the Slack channel or to anyone through email. We are happy to receive your comments and concerns and everything else. So just wanted to throw that out there really quickly. And now I think that I have babbled on for long enough. We can probably get started with the presentation. So welcome, Erin, and welcome everyone. And thank you all for being on this call. And I am excited to see what Erin has to offer today.
Yay, I'm excited to chat with everybody. So today I'm going to be talking about closing genomes with a special emphasis on nanopore sequencing, although a lot of what I talk about today can also be applied to PacBio. So it was really interesting when I was looking for images about closing genomes and circularizing genomes. I found this image of Mr. DNA from Jurassic Park, and this was 28 years ago. So the way genetics was at the time is they were completing dinosaur genomes with frog DNA, which, because we now know more information, that's actually a bad idea. So this is just to remind everybody that genetics is a constantly evolving field, and what I say today may not necessarily be true 10 years from now or 28 years from now. Also, as a side note, Jurassic Park, the movie itself, is old enough to be a bioinformatician, and I thought that was kind of funny. All right, so I'm going to start out with the basics of the basics. First of all, I'm going to be focusing on bacterial genomes. Bacteria still exist, and they still make people sick, even though there's been a lot of focus on COVID. But bacteria in general, they are a single-celled organism that have one large circular chromosome, and then plasmids, which are also circular, which tend to be smaller than the chromosomal DNA. Plasmids are also thought of as being optional, although depending on what species of bacteria you're looking at, some plasmids are required. There's also some plasmids that are rather large and considered superplasmids. We're not really going in depth into that today, but just so as an overly simplistic view, we're using the underlying assumption that we're looking at bacteria and they have circular chromosomal DNA and they have circular plasmids. And we do that, no, that's a little bit more complicated. To the right is a picture of an electron micrograph of a bacterial chromosome, and you'll notice that it does look a little bit more complicated than the simplified picture. We care about DNA because the DNA is the blueprint for what the bacteria can do. So from DNA, we get RNA, and from RNA, we get proteins, and the proteins confer antibiotic resistance and virulence factors and other such things. And so it's useful to sequence DNA because it helps us predict what the protein effects are going to be. This is a screenshot of a CDC website about whole genome sequencing and why we decide why it's a good idea to sequence bacterial DNA. And the overarching goal is using this bacterial DNA to improve public health. So the first point I want to bring up is we use this whole genome sequencing to track and identify what organisms are making people sick. Using whole genome sequencing is a very specific way to track like what kind of salmonella or what kind of Acinetobacter is making a patient ill. I like this phrase of using advanced technology because they've been using this phrase since Jurassic Park. And the MiSeq came out in 2011. So it is kind of advanced and kind of new. Most of the PulseNet sequencing is done on the MiSeq. And I'm going to be talking about nanopore sequencing, which is newer. And there may be more advanced technologies that are going to be coming out later that can also be used to sequence bacterial DNA and improve public health. So this can be used in outbreak investigations. It can be used to learn and predict antibiotic resistance and quite a few more things, which I don't have time to list everything that I can think of that this can be used for. I'm working under the assumption that most of the individuals on the call are familiar with illumina sequencing. So if I'm going to talk about nanopore sequencing, I want to share how illumina sequencing works so that we all start out on the same page of how they're different. So for illumina sequencing, somebody gets sick, a bacteria is isolated from that patient and it's cultured on a dish. That cultured isolate is then, we isolate the DNA from that cultured isolate and we prepare genomic libraries for sequencing. That is adding adapters, which are needed to get the sequences through the sequencer, as well as indexes or barcodes so that we can do multiple samples per sequencing run. And when we prepare these libraries, we essentially take the chromosomes and the plasmids and we break them apart. And so we have chromosomal sequence, we have plasmid sequence, And then we have phi-X sequence. If you're doing illumina sequencing, this helps balance out the red and green lights in the sequencer to give you more accurate reads. And then we throw it into the sequencer, and we get reads from that, and that's great. The reads are chromosomal reads, they're plasmid reads, and they're phi-X reads. So we use a bunch of tools to do some sort of de novo alignment generally, and hopefully that removes all the phi-X. It should remove any stray adapters and barcodes, and we're just left with the sequence, which we can assemble into contigs. Contigs are smaller portions of the genome, and they're not circular. And when we're at this contig stage, we don't necessarily know what's chromosomal, and we don't know what's plasmid. And ideally, we would like to know what's chromosomal and what's plasmid. We want to know, when we're looking at a gene, we'd like to know where it is in the genome. Plasmids are more easily shared among organisms. So if you have like for example, if you have an antibiotic resistance gene on a plasmid, that a little bacteria can share has a better chance of sharing that antimicrobial resistance gene than if it was chromosomally located. And so I was looking for some figures of how to explain why long-read sequencing is better. And there are actually a bunch of really terrible figures out there. And this figure that I'm going to be showing you is from Nanopore. And I admit that it's still not very clear, but man, these are supposedly adapted figures. And if this is the best they can do, there's probably not much else I can improve on. So when we're doing short read sequencing, we get all these short reads here. And when we come upon a problematic region, like this region here, this could be a repetitive region like a microsatellite, it could be a translocation, inversion, something complicated. The short read sequences can't necessarily map across this. And so there's a break, which means that a break here. So this region of the sequence would be a contig here, and this region of the sequence would be a contig here. When you do long read sequencing, that it extends the length of the problem region, so it completely covers it. It suddenly isn't a problem region anymore. So another way to look at this is, so this is up here, we have repetitive regions, and this is the short reads. So if we just look at this short read here, which maps to a repetitive region, in this figure, it's mapped here to this first repetitive region, but it could also be mapped to this second repetitive region or this third repetitive region. We don't actually know where this read maps to. Also, when you look at the non-repetitive regions that are flanking, we don't necessarily know where R and B, that relationship, because in theory, it could be here. But if you're just looking at the short reads instead of the long reads, maybe it goes like RB, RB instead of RB, RC. And so when you're trying to graph these together, you get some really complicated genomes, and they're not as circular as we would hope them to be. When you do long read sequencing, and the reads are longer than your problematic regions, then your regions aren't as problematic as they could be. And so that's this region here is this read is longer than the problematic region. So we know that this here will only map to this region of the genome. And we know that B has to be here on the other side of the region from A. So it helps with mapping and with circularization. Okay, so this is how nanopore sequencing works. PAC biosequencing is also long read, and I don't have a slide for that. But it has some similarities with Illumina MiSeq sequencing and some differences. So I'm not talking about metagenomics right now. So we're still talking about cultured isolates. You take the cultured isolates, you isolate the DNA, you prepare the libraries. So you still have to add indexes, you still have to add the adapters for sequencing, and you still end up with a mix of chromosomal and plasmid sequences that are both shorter than generally what the original DNA sequence was. And then for nanopore sequencing, you sequence it on a nanopore sequencer. And so as audience participation, if you want to type in the chat what nanopore sequencer you have or that you've been using, that would be fun. Although admittedly with full screen, I don't actually know how to read the chat. So you get these sequences and you get these reads, and ideally you align them and you get your chromosomal sequence and your plasmid sequence, and they're all circles and they're all beautiful, and that's exactly what you want. Although I am going to give you this caveat that the long reads have to be longer than your problematic sequences. Otherwise, you still end up with some genomes that are not as beautiful as you would hope. So from nanopore sequencing, you get, so from Illumina sequencing, the raw files are called BCL files. The raw files from nanopore sequencing are called FAST5. And if you do nanopore sequencing, you have to know the difference between FAST5 and FASTQ files. So FAST5 files are the results of the electromagnetic pulses of the DNA strand as it goes through the pore. From the FAST5 files, generally Guppy is used. I know there's a few other tools out there, but Guppy seems to be used predominantly. If you're using Minnow, then there's Guppy under the hood, and Guppy is used to call bases, demultiplex, and remove adapters. And then you get your FASTQ files, which you can do normal bioinformatic analyses with. I point out these two file formats because when you're trying to share these with people, the version of Guppy greatly impacts what your FASTQ files are like. So many researchers and people who are working with nanopore sequencing, they want the FAST5 files. But if you're submitting to the SRA from what I understand, only accepts FASTQ files. This also becomes a really big deal if you are doing metagenomic studies, because since the FAST5 files don't have bases that are called yet, there could still be human or host DNA in the FAST5 files. So there are some concerns with sharing this kind of data. And I don't have a solution for anybody. I just want to point out that there is you may encounter some drama as you're sharing these results with people. So generally the first step is you want to filter the FASTQ reads. So this is a tool, it's called Filtlong. There are many other tools out there, but Filtlong is really easy to use. And I'm going to walk through this graph. So On the left graph is before Filtlong. You can see it as a 2D graph. The x-axis is the read length, going from 0 bases to 1 million. And that is the length of the read. For nanopore sequencing, generally, you're going for longer reads, especially if you're trying to close genomes, because you want your reads to be longer than any of your problematic regions. The y-axis is read identity, which I'm going to use that just as a stand-in for quality. And you want the quality of your reads to be high. And so we'd like to filter out all of the reads that are short. So this is where DNA fragments that are too short to be useful should be in. And we want to filter out things that are low quality. And so in theory, we want to go from a read set that is average quality to a read set that is high quality. The red and blue graphs on the top and side of the graph, that is for height, that shows the density of the dots. So we do see a shift in the density of the dots into the reads after Filtlong. They are higher and they are higher quality, which means we get a better genome. So Filtlong, just as a warning, and any other filtering tool generally cannot filter out all the issues that you're going to encounter. So they are generally bioinformatic tools that are geared toward filtering out bioinformatic issues. There's actually going to be issues in the biology of the samples that you have to encounter. So your jargon word for the day is holiday junctions. If anybody asks you why your sequencing failed, just like start throwing holiday junctions and like other jargon at them. And eventually they'll just decide that you know what you're doing and let you go on your way. Those are portions of the DNA that that they've broken and they haven't repaired adequately. So it's not a linear strand of DNA anymore. So other portions or other problems you're going to encounter is if you're culturing bacteria, evolution is still going to happen. So, and you can't get rid of all bacteriophages, so you're still going to have viruses. Mutations can accumulate, or you can see mutations disappear. Plasmids can get lost while they're being cultured at the lab. And you also get uneven coverage. So what I mean by that is generally people consider like this model when we're using DNA replication. So you take a bacterial chromosome and at the origin of replication, it starts dividing. When we're culturing bacteria in the lab, we have some really great growth conditions. So it looks more like this. So these are all origins of replication at the top, and at the bottom is the termination sequence. So the top here is going to have higher coverage than the bottom here. So you're going to have uneven coverage in your genomes. Additionally, there's going to be some intrinsic properties of the DNA that just aren't going to let it sequence, or that are going to make it less likely to sequence. So now that we've got issues, so I can't solve all your problems, but I'm hoping to solve some of them. So let's say you've done your sequencing and now you want to assemble into a genome. There are a lot of different tools out there. The ones I've named here are Fly, Miniasm, Raven, Canu, Redbeam, Unicycler. There's like Shasta. There's a lot of different assemblers out there. I kind of wish that people would stop generating assemblers, but I imagine that's still going to happen. The assemblers, unicycler is hybrid. Many of them just use the long reads from your sequencing run to create your genome. Sometimes they work great and you get a circular genome, and sometimes you don't. So at this point in time, I'm going to introduce bandage. These two graphs here are considered bandage graphs. Well, and sorry, these are the outputs of bandage. So bandage looks at what contigs are mapped to other contigs. And if you have repetitive regions like this one and this one and this one, and you don't have long reads that span that repetitive region, then there's confusion in the final assembly as to where the contigs meet up. And so instead of a beautiful circle, like this is what, the one on the right is what you want, the one on the left can still happen when your reads don't span your problematic region. So back to long read assembly, I've been asked which one is best. And so these are all tools that are in, there are a lot of tools and they're in a ton of different stages in development. Some of them have very different assumptions. A lot of them have very similar assumptions that they use under the hood. I swear they all use Minimap 2 and Raycon. So they're similar and they're different. So which one is best? And that's when I'd like to introduce Trycycler. So Trycycler is the most like non-committal portion of nanopore sequencing. So Trycycler is not an assembler. What it does is you create multiple assemblies with multiple assemblers, and then what Trycycler is it resolves the differences between them. So you don't have to decide on what your favorite assembler is. You can use all your favorite ones and then use the assemblies that those have been generated to create a final consensus FASTA sequence. So I'm going to be going through the theory of Trycycler and how it works and the steps involved. And yeah. So the first portion of Trycycler is you want to subsample your reads. So we've done a sequencing run. We have all these long reads, and depending on how many reads you have, depends on how overlapping the subsampling is. But you have a bunch of different subsamples. In theory, there are 12, and then they go to different assemblers. So like assembler 1, assembler 2, assembler 3. Doesn't necessarily matter at the moment which assembler you use. Like use your favorite ones that you think are the best for your purposes. So in the Nextflow workflow that I'm creating, I've got Miniasm, Fly, and Raven. They don't necessarily have to be your favorite though. And then the next step is to cluster these assemblies. So each assembly has contigs, which hopefully from the assembly are chromosomal and plasmid. And Trycycler will rename each contig based on what cluster number it's a part of, what contig it is, and then it gives you information about the length of that contig and the depth. In theory, all of the sequences in a cluster should have similar lengths and similar depths. So in this example I'm showing you, this is a very simple example. This is cluster 1. It's likely the chromosome. This is found in all 12 subsets of subsampling. This is cluster 2. It's probably a plasmid. And I say this because its depth is higher and its length is shorter. And it's found in only 9 subsets. So this orange box are all the results from the same assembler. So there are, because of the reads that are given to the assembler and how the assemblers work, they don't necessarily give you the same answer for every input that you give it. So these orange boxes here are the same assembler. You'll notice that the assemblers, they sort of clustered together with similar sequences, but not necessarily. So the next part of the process of the workflow isn't going to work if the length and depth and number of SNPs aren't similar enough. So lengths like this. So the rest of the contigs in this cluster are about 43K base pairs. And then you've got this really nice 87K and this really nice 93K. Those are outliers. And so they're going to mess up the next step. In Trycycler reconcile, you look at the contigs and then you decide if they are similar enough to include in your final assembly. So you're looking at depth, you're looking at length, and you're looking at sequence. So if there's too many SNPs difference, if the length is too different, or if the depth is too different, you have to manually remove those contigs, and those are considered kind of artifacts from the assembly process. And this is why Trycycler, it has a bit of a human touch to it, because the end user has to decide if a contig needs to be moved, or if the depth, length, and mutation similarity parameters need to be adjusted. The next step is called reconcile. So reconcile ensures that all the sequences are on the same strand, that they're similar enough, that they're circular, and it rotates all the sequences so that they start at the same position. The next phase is doing a multiple sequence alignment of all of the contigs in that cluster. So it takes sequences and it aligns them. And I don't really have anything else to say about that. It partitions the FASTQ reads. It goes back to before it was subsampled, and then it assigns reads to the appropriate cluster. And then it creates a consensus sequence from that. So it looks at this multiple sequence alignment, and it notices in areas where it notices that there are differences, it looks at what the majority of sequences have. And at this point, the majority wins. If there's a tie in the assembled sequences, it does go back to the FASTQ reads and uses that as the tiebreaker. So yes, it is all just a popularity contest. And so that's how Trycycler works. It takes a bunch of assemblies, it aligns them together, it notices their differences, and then uses what has happened, like the most common sequence, and then it goes with that. So finally, and I don't have a picture for this. So polishing is when you take steps prior in your workflow, and like files prior in your workflow, and you use that to correct the final assembly. When you do this, you have to be sure that you don't over polish. Many of these assemblers have the parameters and the things that they have because of things they've noticed in their own sequences. So when you use like out-of-date polishers and out-of-date assemblers, you can put errors back into your sequences. So which means that there's no standard polishing procedure. Generally, when people have some sort of assembly, they follow with Raycon polishing and Medaka polishing. And they have multiple rounds of those. Many assemblers actually include polishing steps within them. My favorite polisher is Pylon, mostly because it uses illumina reeds, which if you're using just a nanopore only assembly, then the illumina reeds haven't been included in any part of that assembly process. So they're kind of a orthogonal sequencing that is generally higher quality that you can use to correct errors and things. And pylon polishing, you generally do it in multiple rounds until the polishing results in no sequence changes. This does come with a caveat, though, that pylon polishing is a hybrid approach. So you need your nanopore assembly, and then you need illumina reeds. So if you're only dealing with nanopore reeds, or you're only dealing with PacBio reeds, pylon isn't an option for you. And then if you guys want to join in on the bandwagon, so I'm in the process of creating a Trycycler Nextflow workflow. Because Trycycler has a human requirement in it after clustering and before reconciliation. It's not a complete workflow that takes it from FASTQ reads to final polished assembly. There is a intermission in between, which the end user is actually required to look at the sequences and things. Right now, the workflow isn't complete. If you would like to help me test it out and to add features and that kind of thing, that would be great. It is on GitHub, and I would love collaboration. But in theory, how this workflow is intended to work is you have an initial run into the workflow. So you right now it's set up for nanopore reads, mostly because I don't have any PacBio examples. So we have a grid ion. So in theory, like we have Minnow that took care of all of the base calling, depart coding, and demultiplexing, and we create this directory structure. It reads files in from that directory structure, runs them through Filtlong, the initial phases of Trycycler, to clustering, pauses for a bit where you can examine the tree, manually remove problematic clusters, and then contigs and sequences, and then you restart the workflow for phase 2. And then at the end, you can look at your sequences with bandage. You can take your assemblies to other tools like AMR Finder or other serotyping tools, and you can submit these sequences to repositories such as NCBI Genome, et cetera, et cetera. And there we go. Does anybody have any questions? I'm not affiliated with Nanopore Sequencing, even though I talked about it and I know they're going public soon. Let's see, and I'm going to stop screen sharing so that I can see the chat.
Well, thank you so much, Erin. That was great. I know that there are some questions in the chat. I have a question. It's probably a pretty simple one that we can kind of kick things off. So with Trycycler, are you only comparing 3 assemblies at a time? Is that the max it can do? Is that why it's try in the Trycycle? Or can you compare as many as you want?
You can compare as many as you want. It does depend on how patient you are and what your computational resources are. So yeah, since it is trying to find kind of the appropriate assembly based off of like popular opinion, the more the merrier. Although the more subsampling you do, the more reads that you will want.
Makes sense.
So if you're going to use like 10 different assemblers instead of 3 different assemblers, you should probably have a lot of reads on your run.
Thank you. I think there was a question in the chat about parameters that you use for Filtlong. Do you have a set parameters for that?
So for right now, I just keep 95% of the reads, so the highest quality reads, based off of quality and length. And I've been looking at a couple of different organisms, trying to find optimal coverages and depths that I could recommend to people, and I am not at the point where I can definitively give a recommended depth for the amount of reads that an organism needs. But I wish I did. I was hoping to have it before today, and I didn't.
All right. Are there any other questions?
Sure. This is Taj Azarian. I actually have more of a plug for maybe some assistance with some analysis support. We kind of have a back burner project that's simmering right now where we performed hybrid sequencing, so Illumina and Nanopore on 90 bacterial genomes on Staph aureus. And I assigned my postdoc to do some analysis comparisons between short read only hybrid assembly and then annotation with PROCA versus PGAP, and basically trying to prove myself, like, is it better to do hybrid sequencing? Like, what do we gain? Do we gain better annotation, better ability to look at pangenomes, better ability to resolve nucleotide differences between strains? And we're kind of done a lot of analysis and still kind of at the end scratching our heads. So I feel like I just need somebody else that's played in this area to maybe look through the analysis and the results and see if we're missing something or if there's some other piece that we should be comparing or analyzing. So if that sounds of interest to anyone, reach out to me on the Slack channel, and I can be happy to share our output. But I absolutely love my Nanopore and love hybrid sequencing, and really appreciate the talk. So thanks for the overview.
That sounds great. Yeah, so people reach out on the Slack channel. There's always open communication there. So with the, I guess I have another question about the kind of like manual steps. So you're just kind of like looking at the clusters, and then you can kind of remove or include whichever ones you want in the next phase for Trycycler. I guess I'm a little bit confused about that, Seb.
Yeah, so there are, let's see, I'm going to find one and bring it up. Oh man, I had it right here. And it's not there anymore. Of course. So what happens is what let's see, I will share with you the in the log file, you get a bunch of different matrices that will tell you genetic distance. It will give you length comparisons. And then if you're looking at it in a standard Linux shell, it does highlight in red the samples that are outliers. So if you do try to reconcile with if you do try to reconcile with sequences that can't be reconciled, it will show an error. And it's a kind of painful process to go through because it checks things one at a time, not into the whole thing. So this is from the Trycycler wiki. So here at the bottom, it's got a pairwise identities matrix where it is a matrix, so the samples on the left are the same as the samples on the top. And in that same order. And there are thresholds that you can set. So for like pairwise identities, I think the default is 95% the same. You can increase that stringency, so like they all have to be 99%. And that means if there was a contig that the pairwise identity was less than that, it would cause reconcile to fail. You end up getting an error message. You have to look through the error message and then fix it.
Okay, thank you.
And it is so the getting your contigs ready for the reconcile process is probably the most time-consuming part. And it does require a bit of like, so either the end user has to remove contigs or the end user has to change parameters. So the end user has to decide whether or not that contig should be included or not. But it at least tells you which ones that it doesn't like.
Okay. And then you give it kind of like a list at the end of the contigs that you want to move forward with?
No, so you actually have to remove the files from Trycycler's input.
Okay. Interesting.
Also, since Trycycler does create assemblies through consensus, if you have a cluster that only has one contig, Trycycler won't work on that contig.
So it has to have 2 or more?
Yeah, I think it has to have 3 or more, actually.
Well, that is really interesting. Yeah, I'm excited for Donut Falls and for people to start kind of utilizing this and working with some of the long read data that is coming out of different instruments. I guess, I mean, I know that a lot of people have been using their MinION instruments, but I wonder, has anybody used anything that uses like a MinION on the back end for this kind of data? Oh, it looks like Curtis, you raised your hand.
Hey, yeah, I just have a quick question. Awesome presentation, by the way, Erin. Really enjoyed it. I'm curious what tools you are using to generate the assemblies that go into Trycycler, like what you've experimented with.
So I've experimented with the ones in the Trycycler tutorial plus Canu, because the ones in the Trycycler tutorial is as I was getting Trycycler working, right? And then Canu because there was already container for it on StaPH-B. I have not tried Canu 2. And so right now in the workflow, it's just Miniasm, Raven, and Fly, because their containers worked and they were nice and it was great. Canu, when you have really, really long reads, Canu takes forever. And so I let Canu run for like 2 days and it still wasn't done with 48 cores. And so I killed Canu and I haven't really looked back.
Got it. Thank you.
But not saying that those are like the best ones or that those are going to be the final ones. Like I really like how non-committal Trycycler is. It's just kind of, use whatever assembler you like, and then it will create the final consensus through kind of popular vote.
So I will just mention who was that? Thomas Christensen added the Canoe 2.0 Docker file. He said to the StaPH-B repository. He said that it was much faster than V1. But I also remember like way back when I first tried Canoe, there's an input parameter you have to use in order to, I forget if you have to give it an estimated genome size, or maybe it's like a read max number of reads to use.
Give Canu a genome size. It also does have a fast option, but the quality goes down significally if you use that. So
yeah, I just remember trying to assemble like Lambda with the minion test data, and like it took hours, and it was because I forgot one of those parameters. So I don't know, maybe that's it, maybe not. But
no, I gave it the genome size, and it, yeah.
Gotcha.
Like if it can't do one genome over the weekend, like I can't use
it. On to the next assembler.
Yeah.
All right, well, thank you.
So I know that there was, you had some questions where people were confused with unicycler and Trycycler, but so like theoretically, could you use unicycler in Trycycler?
Yeah, so in theory, you could put, so all Trycycler really cares about is like you subsample the reads and then you get an assembly. So yes, you could put unicycler into Trycycler. You could put any kind of assembler in there that you wanted to. Although if a config can't be circularized, it does cause reconcile to fail. So I don't recommend just throwing short read contigs in there. I don't think that would be a good idea.
Good to know. All right, are there any more questions? I know that we're coming up on the hour, and I know a lot of people have calls back to back after this. So I want to get, make sure that I get all the questions and everybody's concerns addressed. All right. Well, since I'm hearing nothing, I know that if you do have questions, you can reach out through the Slack, or I think Erin will probably be more than happy to answer emails if you email her.
Yes. Or through Slack or through Twitter or email.
There's multiple ways to get in touch. But yeah, so I guess if there is nothing else, I want to thank Erin for putting together this presentation and agreeing to speak on this month's call. It was really awesome to get back to some of the kind of, core bioinformatic tools that this group kind of started out with. And so it's an exciting development to get back to that. And yeah, I am really looking forward to seeing how people do with this new tool and see if it actually works out for you and if you are able to kind of reconcile some of your long read sequencing and get some good assemblies. So thank you everyone for being on this call. And I'm going to go ahead and stop the recording.